This is Chapter 5 of the Instructor's video manual for cloning a fluorescent gene. By this time, hopefully your students have lots and lots of colonies on their agar plates that they plated after transformation in Chapter 4. Under UV light, about 50% of these colonies should be GFP expressing, and about 50% of them should not be. This is because the insert that we ligated into our vector construct can either go in forwards, where it reads in frame, creating a successfully folded GFP protein, or it can insert backwards. And then when the promoter creates mRNA from the reverse GFP protein, it ends up being gobbledygook and doesn't show up green. Today's lab should be really fun. The students have now learned how to use biotechnology equipment and gone through all of the procedures done in a real biotech lab to clone a gene. And they've seen now the result of that process. So Chapter 5 is about a really fun activity where the students get to take their successfully GFP expressing transformed bacteria and draw a picture of their own design for it. The details of that are in the lab manual, but we wanted to give a couple of quick tips for how we like to do it here at Gen.otyp. The first trick that I want to give you is having the students trace out a Petri plate on a piece of paper and draw their design there, so that then when they come with a Petri dish, they can just put it over their design and use whatever tool they choose to make their mark painting with bacteria. Our favorite tool for painting with bacteria is the pipette tip. You simply mix it around in an Eppendorf tube full of your bacterial paint and then dip, poke, 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 almost like those punch-out cards that we used to do back in like kindergarten. So poke, 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 poke on the plate with the Petri dish sitting on top. Kind of like that. Now, if by some chance something went wrong on our end or something went wrong on the student's end and you guys have no GFP colonies, don't panic, because we included the control plate, which has GFP colonies from the tests that we run to make sure that all of the reagents that we send you are working before we send them. So we send a plate that has GFP colonies on it. That means even if your students' experiments don't work out, you can let them use this control plate so that they can still get to do this fun activity with the glowing bacteria.